Dear learners, in continuation to the system of classifications, the next system of classification is taxonomical classification. The term taxonomy derived from the word taxis, which means the arrangement. Taxonomy is the branch of science concerned with the classification of organisms. So in this system of classification, the drugs are classified according to the plants or animals from which they are obtained in phyla, order, families, genera, species, subspecies, etc. This method of classification is based on the consideration of the natural relationship or phylogenetic arrangement among the plants or animals. The crude drugs of the plants are classified on the basis of the accepted botanical system of classification. In here you can see the example. The phylum is Spermatophyta, the division is Angiospermi, class is Dicotyledonous, order Rosalis, family Leguminosae, genus is Glycyrrhiza, and the species is Glabra. So this is the classification system of Liquidis, common plant Liquidis, or Geostimodhu or Mulethi. The major merits of this system of classification, it is very essential for the botanical description of the medicinal plant, because most of the taxonomic system of classification is based on the botanical arrangement, the structural arrangement of different organs, and indirectly useful for the morphological identification. For example, Umbelliferae family or plants generally bear the cremoca fruit. This system is also essential to reduce the confusion between the vernacular name. So if we know this binomial name which denotes the genus and family, we can easily reduce the confusion of different vernacular name of the same plant. However, this system of classification, although looks appealing, but many drugs are not entire plants and represent part of the plant that have been processed systematically. This system of classification also fails to take into an account of the chemical nature of active constituent and therapeutic significance of the drug. The next system of classification is chemotaxonomical classification. This system is very important system of classification because in this system of classification we consider both the taxonomical arrangement and taxonomical positions along with their chemical constituent or the secondary metabolite. In this system of classification, we mainly focus on the secondary metabolites and their systematic arrangement in nature. The drugs are classified according to their chemical constituent and their taxonomic position in nature. This system considered the phylogenetic relationship with the secondary metabolite genesis. So previously in chemical system of classification, we mentioned that the primary metabolites are the essential for plants' growth and development, whereas the secondary metabolites are the byproduct that derived from the primary metabolite. And the amount of the secondary metabolites or the availability of the secondary metabolites is actually restricted to the different taxa, not the universal for all the plants. For example, Solanaceae family plants contain tropane alkaloid. Umbelliferae family plants contain volatile oil. The merit of this system of classification is it is essential for the new drug discovery process, and it also gives an account of the how the secondary metabolites form in different taxonomical arrangement. Demerits are this is highly sophisticated technique and required DNA hybridization, amino acid sequencing, and other essential tool to express. Sometimes it's also difficult when some geoclimatic effect applies on the plant to establish the chemotaxonomical relationship. Next system or last system of classification is serotaxonomical classification. It is quite newer system of classification. Serotaxonomy can be considered as an application of serology on taxonomical classification. Serology is the study of antigen-antibody reaction. This reaction may be the same or may be different in different taxa. On that basis, drugs may be classified without considering their morphological or sometimes phylogenical relationship in taxonomy. The classification of very similar plant which cannot be distinguished on the basis of the taxonomical classification or chemotaxonomical classification can be distinguished by the means of differences in the protein they contain. So basically in the system of classification, what we do, we actually collect the protein or extract the protein from the plant, which is injected into the bloodstream of animal, that may be a rabbit or rat or other animal. When that protein, the foreign protein, plant protein is injected in the animal, they behave as an antigen, and on response to this antigen, the animal body produces certain antibodies, and that antibodies are collected by collecting a blood sample. Then we compare this protein, antibody protein from the first plant protein with the extract taken from the other plants. So the nature of the antibody produced by the animal is different from plant to plant. On that basis, we do the classification. The advantage of this system of classification, it helps in the comparing non-morphological characteristics, which helps in the taxonomical data. This technique is also helps in the comparison of the single proteins from different plant taxa. However, the demerits are like the chemotaxonomic classification. This system of classification requires sophisticated instrumental support and not beneficial for the phylogenetic establishment, because we don't know that which protein comes first in a plant's body. So this is all about the different system of classification. Thank you.